Ok, então já está.
Hi, everyone. Hi, good morning. Thank you for joining us in another CNC meeting online. Thank you for coming to this session. We will today learn a little bit more about the work that is done in the microscopy facility of the Faculty of Sciences of the University of Lisbon. We will learn that it is not just for the small stuff. With Luís Marques, researcher at the Center for Ecology, Evolution and Environmental Changes, to whom I thank accepting our invitation for this presentation. Before I give the floor to Professor Solveig, who will also say a few words about Luís Marques and about this topic, I invite you during the session to leave your questions in the YouTube chat, which you have on your right on your screen. So you can leave your questions during the session, and we will have some minutes after the presentation to ask these questions to Luís Marques. So without further delay, I now give the floor to Professor Solveig, who I also thank being here. Thank you. Oops, your microphone.
Yeah, my microphone. Thank you very much, Marta. It's a great pleasure to introduce Luís Marques, who I've known for 20-some years. He took his biology degree here in FCUL and did the ramo científico, as it was called at that time. And after he finished his 4 years, he did a 5th year at the University of Amsterdam, working in the Netherlands Cancer Institute in hardcore molecular and cellular biology. When he came back, he joined our group as a research assistant and learned a lot more about microscopy than he had learned in Holland. This was at the time when Gabriel Martins was a postdoc in our group, and he brought lots of knowledge about bioimaging from his PhD in the US. So Luís fell in love with microscopy very soon. So he will tell you the history of how the microscopy facility grew and his role in it also, which is a great example of how a bottom-up approach can end up in a facility some years later. So Luís very quickly was always helping everybody around him in their problems of microscopy and image processing, and soon found his calling, let's say, which is basically helping others to set up their experiments and get the most out of their data. So when the opportunity came to become part of this service provider in the facility, as a service provider in the facility, he was, of course, first in line and has been working there since. And so I think we should give the word to Luís to explain what they do and how they came about to do all the nice stuff in the facility.
Thank you very much, Professor Solveig. Can I start? Okay. So good morning, everyone. Good afternoon right now. Thank you very much for having me. It's a pleasure to present an Encontro Ciência a few years later. I'm going to talk to you a little bit about the FCUL Microscopy Facility. Right now, I am located at one of the rooms of the microscopy facility here at FCUL. I'm going to start my presentation. One moment, please. Let me just switch this. Okay, can everyone see Marta and Solveig? Is it okay? Okay, so I'm going to start by talking a little bit about the FCUL Microscopy Facility. Right now, I play the role of optical microscopy technician here. I belong to both C3C and to BioEasy research units, and that's essentially the story of the facility. Right now, it's an infrastructure of FCUL that is sustained and composed by both these research units and aggregating all of their different competences and equipments and know-how in providing service in microscopy and in image analysis to the FCUL campus and beyond. So this here in my cover image is a small screen capture of the FCUL Microscopy Facility website. You have all of this information about the facility. You can come here. It's at fculmf.campus.ciências.ulisboa.pt. Please feel free to visit us. There's a lot of different information here about us, about the equipment, about how you can do your work here. But for the time being, I'm going to talk to you about the history of the FCUL Microscopy Facility, where we come from. So as I said before, it's actually a joint infrastructure of both these research units, CE3C and BioEasy. And we serve as a service provider for microscopy and image analysis, and also for training. We play quite a big role in classes, as I'll tell you a little bit later today. But we started more or less in the late 20th century, in the late 80s, early 90s. In the 80s, FCUL came to Campo Grande, and we were supposed to bring along both our electron microscopes, but unfortunately that was not possible due to technical issues. But the researchers that eventually formed the CE3C predecessor, the Centro de Biologia Ambiental, the CDA, were able to acquire a scanning electron microscope, which is still in working today under the expertise of our electronic microscope technician Telmo Nunes. Also in the early 90s, the Departamento de Biologia Vegetal acquired the first confocal laser scanning microscope in the Iberian Peninsula. It was a Biorad MRC model, which is no longer working. It's in parts, and I'm actually looking at it right now in our museum room, where we have all of the equipment that was deprecated, and it serves as a reminder of how things of the old equipment that we used to use, but since technology advances, we started to have to put some things aside. It's still a very beautiful machine that greets everyone that comes here to visit the facility. In the 2000s, there was an interest to aggregate all of the different microscopy equipments present at Department of Animal and Plant Biology into a single lab, room 2115, which is where I'm located right now. We have part of the optical microscopy here, and also the electron microscope, the scanning electron microscope. And also in the 2000s, the Carlos Gulbenkian Foundation was of paramount help in the acquisition by both departments of a Leica SPE confocal microscope, which is right there to my right. I won't be able to show it to you right now, but I will show it later. And this was the consolidation, more or less, of the FCUL Microscopy Facility as a facility with actual equipment and integration of knowledge to provide to the community. In the 2010s, we took another step forward, since the old CBA and the old BioFig research units evolved into C3C and the BioEasy, the modern incarnations of these research units, and expanding their scope. Also, BioEasy in 2013, I believe, acquired the first high-throughput wide-field microscope in the Lisbon area, and these days we are essentially one of the main high-throughput microscopy service providers and researchers here in Portugal. And also during the 2010s, we were able, both BioEasy and CE3C were able to take advantage of FCT's Roteiro Nacional de Infraestruturas to update a lot of the equipment that we have here at C2, and also to purchase a Leica SP8 confocal equipped with fret film technology. Also during this time, since we were expanding our scope as well and acquiring more equipment, we set forth the creation of the FCULMF website and to constitute the facility as an actual Faculty of Sciences infrastructure. Also, during this time, since we were consolidating everything, we also adhered to the Portuguese platform of bioimaging. And then recently this year, I believe, we were accepted through the Portuguese platform of bioimaging into its European counterpart, which is the Euro Bioimaging Network, with access to plenty of resources and technical know-how, and some of the top researchers in microscopy and image analysis in Europe. They are available to us and to collaborate, since we belong to this network. Who we are. So we have Professor Luís Marques. So this is a joint venture between C3C and BioEasy. We have Professor Marques from BioEasy as the facility head. We have Hugo Botelho, who does his work at Professora Margarida Amaral's group at BioEasy, and he is the head of the High Throughput Microscopy Facility. I will talk a little bit about the work that they'll develop a little bit later on. We have Professora Solveig and Gabriela from C3C that are part of the Coordination Commission. And then we have two technicians, both Telmo Nunes, who works at the electron microscope, and also myself, and I'm the optical microscopy technician. The rest of this presentation will actually be very focused on optical microscopy. Since I'm sure that Telmo will be happy to present some of the work that's done on the electron microscope. And I believe that C3C as a community is a little bit more aware of the electron microscopy here at the facility than of the rest. So I will try to talk a little bit more about that, and to present all of the installed capacity and equipment and know-how that we have on optical microscopy here. So we're composed of two subunits. So we have the FCUL Microscopy Lab, essentially this place where I'm stationed at C2. We're dedicated to essentially morphological analysis. Using stereo microscopy, wide field microscopy, fluorescence, confocal microscopy, electron microscopy. But we also have at the C8, with our colleagues from the biochemistry department, we have the high throughput screening facility, which is dedicated to essentially high throughput screens. I encourage you to go look at Hugo Botelho's presentation of the BioEasy high throughput screening facility. It's available at YouTube with this by searching these terms. So here Hugo is showing all the different equipments that are necessary to try to analyze a great amount of biological material, usually cells, which are analyzed with motorized and automated microscopes in order to try to assess different morphologies, different phenotypes, and essentially they work a lot in cystic fibrosis, trying to pick out genes or compounds or pharmacological strategies that may ameliorate this disease, and try to come up with therapies that may improve the life conditions of cystic fibrosis patients. So I encourage you to look at this, to go watch Hugo's presentation. But I'm going to talk a little bit more about more or less the morphology aspect of this. So the microscopy lab, where I'm at right now, if you want to be localized, so right there is these bottles. So because of COVID, we have to take great care in disinfecting all material. Right now, we're not accepting any work that's not very urgent. But when we were accepting users, we enforced very restrictive disinfection rules of all equipment so that we can keep all of us safe. So right now I am working at this computer right here. It's one of our 3D analysis workstations that we have in this room. This is the other one. We also have an upright fluorescence microscope that's connected to a color camera. This is usually used for histology and for color acquisitions. Here is our confocal microscope, which is also equipped with a camera, so it's also a wide field system. Right behind me, it's this room, room number 2, where we're equipped with a couple, this one and this one, we're equipped with a couple of fluorescence microscopes. This one is inverted and is equipped with micro manipulators, and this one is a regular upright fluorescence microscope. Here is a fluorescence stereoscope, which is used for bigger specimen. And here, for field samples, we have a computer-controlled SLR, digital SLR camera. And here we have a couple of 3D printers that we essentially use to make components for microscopy, but also for other situations that may be necessary to have really established shapes and components that may be used in fieldwork, for example. Right here, I don't know if you can see, but there are a couple of adapters that I may show you later on that are used to couple smartphones like these to system eyepieces to capture images and to aid in classes. So at C8, we have the high throughput screening facility at rooms 8171 and 8179. At this room, at room 79, we have that wide field microscope that's used for screening. So it's a fully motorized system equipped with an incubation chamber so that we can do in vivo experiments with live cells or with live tissue. We also have here at the bottom are the equipments necessary to do the proper treatments for culture plates, in which we have all of the cells that will be analyzed automatically at the microscope. And we also have 4 screening analysis workstations, which are located here to be used by the researchers. At room 71, we have our, it's essentially our Ferrari. We have a confocal microscope. It used to be the latest model from Leica, but it's very, very recent. It's equipped also with an incubation chamber to do live experiments, and also with Fred film technology. And it also has the same screening capabilities as this one. So these are equipments that are used essentially for high throughput microscopy and screening. So our output here, I will show you. It will be shown some pictures of the different equipments while I tell you about the output. So in 2018 and 2019, I think everyone understands that our usage scenario has been very different throughout 2020 because of the pandemic. But from 2018 to 2019, we served 82 internal school campus users from the research centers that compose the facility, BioEasy and C3C, but also from Instituto Dom Luís, from MARE, from the departments, and as well as from the Museu de História Natural. Also, we have external users from different from different institutions in Lisbon, but also from the Açores, from Algarve. We provide these services for all of these institutions. We have at least 12 peer-reviewed research publications that are directly related to work done at the facility. I say at least 12 because this number is not updated. We have more right now. We did a couple of workshops on both high throughput microscopy and epithelial systems. And also we helped at multiple animal biology and plant biology departments, and also to Departamento de Química e Bioquímica undergraduate courses. And we also have a dedicated bioimaging subject that's lectured by Gabriel Martins, as Solvik was mentioning earlier. Telmo Nunes also used to welcome students from high schools, and he showed the electron microscope. So this is all of the different usage case scenarios that we have here at the FCUL Microscopy Facility. So how can you do experiments here at the FCUL Microscopy Facility? So first of all, as I mentioned previously, go to the FCULMF website. It's this address right here. Go to user sections. That we have a lot of information there and instructions on how you can be a user of the facility. Since we have a booking system in place, it's the Agendo booking system that was developed by researchers at the Gulbenkian Institute of Science. And they gracefully let us use the platform in order to register and account all use of the microscopes, and to ensure that we have a convenient way to book the systems, and so that the users don't overlap in usage. You have the register button here if you want to create an account. And now the issue of cost. So these imaging facilities, it's always a sensitive subject, but imaging facilities are very expensive to run and to maintain. The upkeep is very high. We are working with systems which were very expensive to acquire. And unfortunately, they're also very expensive to maintain because they have a series of consumables like fluorescence bulbs, which are cheap, a few hundreds of euros. And then we have lasers, which are not cheap, and they're tens of thousands of euros to replace. So we are funded by the strategic funding of both CE3C and BioEasy. We are funded yearly, and the amount that we receive is usually enough to maintain the facility and to allow the usage from internal users, from BioEasy and from CE3C. And I say that it's usually enough if nothing breaks. So if nothing breaks, we manage. But things do break, and it's equipment that is very, very sophisticated, and if you use something that's very sophisticated, you are bound to damage it. And sometimes we do have to send equipment for repairs. Some parts have to be replaced, and these parts are unfortunately very, very expensive. So as such, all the usage right now is accounted through Agendo. And users, if the need arises, will be charged according to the proportion of the time that they used. So it's like this: if your use of the facility is quite sporadic, every once in a while, sure, we'll manage. There will probably not be any costs, but every once in a while, things break that have to be replaced. Computers break down, which have to be purchased. Again, some parts have to be replaced. Some software has to be updated. All of this has costs, and every once in a while, we do have to charge people. So if you're planning to do extensive imaging and analysis, we kindly ask you to do two things. When you're submitting projects under FCT calls, please check estimated use and pricing with us, and budget an appropriate amount of funds for microscopy in your project, in your call. This will make your project more sound. FCT evaluators take this into account. They know that all of the work that's done in microscopy and image analysis, it has costs everywhere. So this will make your project perfectly in line with everyone else's, and this will be an asset. Also, always provide this institutional information, because we have other sources of funding, in this case, the PPBI, the Portuguese Platform for Bioimaging, which has provided us with funds to do some updates of our equipment and to purchase the C8, SP8 confocal, and also as of now, for the time being, is paying my salary. So it's always good to, if you publish, to mention this information so that we can keep being able to applying to funds and to these kinds of initiatives. So why do we have such sophisticated and expensive equipment? Why do we need it? Isn't a microscope just like a set of lenses and a set of eyepieces so that we can look through? Well, actually, the problems in optical microscopy is that it's quite difficult to observe properly biological samples, because you have here a couple of examples like human hair, and this is a focus. A Z-stack of moth genitalia. You can see that we have a couple of problems here. So for instance, the tissue is quite dense. We're not able to see what's inside of this human hair, of all of these structures. We can only see what's in this case, the focal plane, and we can't see inside. In this case, when we have thick samples, we have access to a lot of different focus planes, so it's difficult to capture a good, proper view of the structure that we're trying to see. And how do we usually solve this? We usually solve this, and we're used to doing this since the early days of everyone's life microscopy, like in high school. We used to get really thin samples, like using very fine blades to section parts of biological material, or looking at monolayers of cells like we have here. These are CHO cells in bright field microscopy with no contrast, and you can see what the problem is. When we get really thin samples, we have very, very little contrast, because biological material has a lot of water. Cells are like bags of water with stuff dissolved inside, so there is very little contrast, and we have to employ a series of stratagems, some of them part of the microscope machinery, others involving dyes or stains that will create contrast inside cells. And when we do that, when we have specific technology that allows us to, let me just start all of these videos. When you have access to specific technology that allows us to work around these problems, like thick samples or lack of contrast, we get images like this, which are essentially produced by very sophisticated machines. So here, for example, in this one, we have this is a mouse embryo, I think 13.5-day-old mouse embryo, which was sectioned from top to bottom, and each of the sections was microphotographed. And then we used specific software which aligns all of the different slices and allows us to reconstruct the whole volume. Of course, this software is also paid, it's also very expensive, but this is the kind of things that we can do when you have access to proper technology. Here we have a muscle bundle, also a mouse muscle bundle in one of these embryos, which we stained with fluorescent antibodies, which allows us to look at proteins inside the cells. So here we have a signaling protein called AKT, and we have a marker for myosin, so which is one of the contractile proteins inside cells. And here you can see the entire muscle bundle and also the spinal nerve that innervates this bundle. This image is a confocal stack, which was acquired at our confocal microscope. Here is another example of that. So this is the dermomyotome. It's one of the precursors for the muscles in embryos. And here is an actual, the early formation of muscles in the myotome. So this is the precursor for the cells that will later form the muscles. And here, so this is technology that allows us to do, so these are physical sections of material, and this technology, optical confocal microscopes, allow us to do optical slices of biological material. Using very sophisticated machines, we're able to look inside the tissue. But we have to use equipment that's hundreds of thousands of euros to capture these kinds of images. And also here we have an example of optical projection tomography. This was done by Gabriel Martins here at FCUL. He constructed one of these microscopes, which enables us to get images and inside images of bigger specimens. So this is a newly hatched chick, I believe, and we can see inside. But this is the kind of technology that has to be used in able to have these kinds of images of biological material. It's fairly expensive to be able to get these kinds of images. But one of the advantages, as you can understand, all of this are digital reconstructions, are 3D reconstructions done in the digital world. Most of you may know the story of Nicholas Hartsocker. He was one of the early microscopists. He was a very talented microscopist, but at the time, he could have sworn that his his observation through the microscope of samples of sperm, since he was informed at the time by the preformist theory that all of humans were preformed in sperm, he could have sworn when he published his essay, the Dioptrique, he could have sworn that he saw a whole man, a small man that only had to grow outside during gestation. He could see the whole man inside the sperm. So of course, the advent of cameras, at first film cameras and then digital cameras, enabled us to bypass some of this subjectivity. And these days, modern imaging sensors like cameras or photomultiplier tubes capture images of the biological world as matrices of ones and zeros. So a representation of the real as a digital file, which is composed of pixels, and all of these pixels are zeros and ones to which we can apply mathematical operations and do quantification. So it's not just for morphology and for archiving of structures anymore. It's also for quantification. And this is the part where I start to bring some of the eco and evo biologists into the mix. So we were talking about the microscopy world. We were talking about cells. We were talking about tissues. We were talking about embryos. But image analysis is something that can help any biologist, and we have tools for field biologists as well. So this, for example, this example is a very simple, very simple example of what we could do. Let's assume that we had a video of a flock of birds and we wanted to count how many birds were present in this single frame. So we can use a tool called Fiji. So Fiji is a distribution of a program called ImageJ, and Fiji is the distribution of ImageJ that's most used for biologists and people working in biological sciences all around the world to do processing and analysis of biological data. So I did this very crudely in like 30 seconds. I wanted to be able to count how many birds were in this image, and I recorded myself doing so in Fiji. And so what we usually do it's force of habit. We always duplicate our original image so that we don't change it. Force of habit always to work in a grayscale, because we are going to try to segment, to extract information of this image based on the gray level of the pixels of this image. I'm going to do some pre-processing, a simple filter to blur the image a little bit so that each of these birds becomes like a blob. And then I'm going to use a simple tool that's going to detect the darker pixels in this image, which is called fine maxima. In this case, it's a fine minima, since we're using a light background. And then we change the preeminence, so how narrow or strict this count will be. So this is a preview of all of the maxima. You can see that some of them are not being caught. That one is a little bit too much, so I have to change it. So here we go. This is a very simple and very crude way of assessing more or less how many birds would be in this frame. So in this case, it would be 1,064 birds. I mean, you can understand that this is very simple, because some birds may be on top of one another. We have little clusters here, so this is not perfect. But this is something that I did like in 30 seconds. When we do this properly, as with any biological protocol, things have to be really well thought out in advance, and we have to take good images. And these days, even with smartphones, we get really good images if they are properly taken and properly calibrated. Even with smartphones, we can do some sort of image analysis and some sort of quantification. Remember this example that I showed you previously, like the moth genitalia, the way that we have a lot of different focus planes. So there is one of the plugins of Fiji. We can do what's called a focus stack. This will be very familiar to people that do macro photography and do different Z steps in your focus, and then assemble all of the focused points into a single image. And that's what we have right here. As you can see, there are some artifacts here given because of the out of focus information that is still part of the image, but still for some uses, this is very, very useful to be able to look at the entire stack completely focused. This is another very simple, very crude example of what we can do. But now for something a little bit more sophisticated, something that we have been working on with the lab of Sara Magalhães. So they work with spider mites, and they were trying to assess how much foliar damage was being done in leaf arenas of specific plant species. So this is quite difficult to do. One of the things that is usually done is we draw a physical grid on top. We place a physical grid on top of the plant, and then just check all of the different squares in which damage is present. But if you have a lot of replicates, this is completely this is not feasible in proper time. So this experiment is also done with multiple variables. So fitness assessment, the mite type, density of infection, type of plant, hours of infection, and the replicates. So what did we use for this? So you remember when I'm going to go back a little bit. Remember where I showed you here that I was trying to segment this data based on the gray level? That is the simplest way of segmenting an image based on the tonality of each individual pixel. And we could more or less do that here. We could also try to assess the tonality either of the color or the gray level of these parts with the damage, and try to segment that out of the full image. But these leaves and their lab's leaves had a problem, which was the veins. The plant veins here were actually more or less the same color as the damage. So we would be overestimating the amount of damage. So we had to use another approach, which was, first of all, properly acquire and name the images. We had a lot of different images. These ones were first taken at our stereoscope in quadrants, which were then stitched in Photoshop. Photoshop is not your friend. Mostly, a lot of people do some image processing in Photoshop, and a lot of the times they use it to commit scientific fraud. So Photoshop is not your friend. In this case, I had to use Photoshop to do proper blending of image quadrants, but it's best to use another kind of tool that is more established and has proper protocols to be able to analyze scientific data. So we had all of these samples. These samples had to be pre-processed and cropped in order to be analyzed. And each of these files is unique, so that we have access to all of the information from the replicate right here. And then we use a program called Elastic, and this program does machine learning. So this program, we create different classes of segmentation. For example, in this case, for out, for everything that's not damaged or leaf. For the rest, in this case, the healthy mesophyll of the plant, and also for the damage. We literally paint on top of some of these images. We train the program to be able to recognize the damage in, and then to later on to apply this learning to a folder of images. So in this case, we had 1,150 instances. So that were, and we used around, I believe, 13 instances of training to be able to detect damage in all of them, and try to keep the plant veins out of the analysis so that we wouldn't be overestimating the damage. So the final result is this. We have in red, we have the damage, and then we can use a simple macro in Fiji that I will show you an example later on to assess the area of these red parts. And then in Excel, we just divide this measured area by the area of the full arena. And this will give us the percentage of damage. So this is one of the ways that we can use image analysis in one in an Evo-Devo context. Another one that we had here, I was helping a colleague from BioEasy. She wanted to assess. She was working with plant hypocotyls, and she used a specific fluorescent dye to mark suberin, and she wanted to assess in different instances what was the area of the whole hypocotyl, this one here, what was the area of the xylem, and what was the area of the periderm, this outer layer here, and try to assess relative fluorescence of this dye for each of these measured areas. Now, let me make a disclaimer. It is fairly difficult to assess, to quantify amount of fluorescence in microscopy images. You have to use you have to take all of the images with exactly the same settings, and you have to do lots of corrections and normalizations and controls, and even so, it is quite challenging, but it can be done. So in this case, I established a Fiji macro that would be able to segment all of these structures and then compare all of them and export this data to an Excel spreadsheet to be able to compare. And this looks a little something like this. So this is steps 1 through a lot. And essentially what this macro does is take one of these files, one of these images, and does the following. So I saved this this protocol as well. So we're going to start the macro in this image. It's going to trace through all of the different steps. It's starting the segmentation. It's starting to, you can see here, a results table with measurements, and it's going to acquire more measurements. Right now, it's trying to segment the part of the xylem. And in the end, we'll have a fully segmented file like this. And these are the end results. You have the original file and a fully segmented file and a results table with the area for each of these structures that we want to segment, and absolute or relative values for the amount of fluorescence in each of them from the dye that is standing for suberin. And now I just sent all of these images to the colleague, and she will start crunching all of this data from, I believe, 300 different hypotheticals in this image. So this is another thing that we can do with image analysis. Some things that I would like to do, future prospects for the facility, and to also help the work of field biologists. So I don't know if you know what photogrammetry is. It's a technique which involves taking pictures doing a cloud of pictures at different angles of physical material, as you can see here, for example, and we rotate the material and we construct a cloud of photos that we can use specific software to generate really detailed 3D models, and there is quite some use for entomology and for insect libraries. So this is one of the things, these are one of the tools, since we have a 3D printer, this is one of the tools that can be printed in-house and to motorize with simple stepper motors that are available in robotics shop. We could do this in-house, and I was really thinking of doing one of these things, but there have been some developments with a lot simpler systems, like using modern smartphones which have depth sensors, and this facilitates a lot the acquisition of 3D models from physical material. So I'm starting to investigate a little bit more on the feasibility of a simpler system like this, and eventually I will give you some updates on this. And I also count on your feedback to see if this would be something that could be useful for people at C3C. I would also like to do to install one of Gabriel Martins' OPTs here at FCUL. So he constructed this first one here, and I would like to construct another one as well. And this is one other thing that I would like to, another tool that I would like to have access to be able to image bigger material, like, so that we are very, very limited with the size of samples that we can put in a microscope or even in a regular stereoscope. So this would allow samples and specimens in the centimeter range, which would be a very useful tool. And that's about it. So some of the final remarks. Bioimaging is a powerful tool when studying the living world, but we are not stuck anymore with simple specimen or phenotype archiving. We can also use proper digital images, which are properly acquired and calibrated to do analysis and quantification. And it's very likely that even data from field biologists could benefit very much from image analysis. And if you're interested in doing so, please come talk to us. And that's it. Thank you very much for your time, and I'll be happy to take some questions.
Thank you very much, Luís. Thank you for your very interesting presentation. Congratulations for your work. And as you say here, bioimaging is really a powerful tool, and it's impressive to see to how many different projects you can use it with to help us learn more, and lots of exciting projects ahead. So we are looking forward to hear again from you in the near future. We have now some minutes for questions, if there are any questions. I'm checking the chat, and you have actually lots of people congratulating you. So so everybody is congratulating you for the presentation. In the meantime, there was one question about the website that you mentioned in the beginning for the microscopy facility. In the meantime, we have included it here in the chat of the YouTube, and it's also in the description of the YouTube video. So if you have any questions, please leave them here in the YouTube chat. In the meantime, I also asked Professor Solveig, who is also with us, if she has any comment or any question about this work and about prospects for future research. Let me see.
Yeah,
Professor.
here.
Yes.
I think it was clear from Luís's presentations that this technology has applications for many different things. It's basically you can measure and reconstruct and with these protocols, you can even make quantitative measurements of all kinds of scales, and very often we don't think that it's possible to look inside an insect, you know, without cutting it up, and that is basically what things that you can do. Of course, people that work on the micro scale are used to using microscopes and these types of technology, but people that use intermediate scales perhaps are not as familiar. They're more familiar with satellite imagery, and which is the other extreme of in terms of size of samples. So I think it's Luís loves helping people. So I think if you have any problems or any issues that you would like to discuss with him, he will certainly find you a solution, or try at least.
Thank you very much, Professor Solveig. And in accordance to what you were saying of Luís also always being available to help people, we have here, among the several comments that we have here on the YouTube chat, Marta Santos, an anonymous of mine, she also says, thank you for the cool and informative talk, and also for being always so available and helpful through the years at the MF. So thanks, Luís. So we don't have questions for now here on the
Sorry, if I may, Marta.
Yes.
If I may, regarding Marta Santos, Marta, the work we're trying to detect the Drosophila eggs, it's not forgotten. I was just very overwhelmed by both these projects that I showed you, and also by a recent birth of my daughter. So, but that detection of eggs in Drosophila is not forgotten, and I will try to do my best to work on that as well.
Thank you. That's great. So we have here an exchange of information about future projects. So projects ongoing. Thank you, Luís. So we don't have further questions here on the YouTube, but if you want, you can also contact Luís Marques afterwards. You can find his email and his contact on our website as well. Marta Santos on the YouTube chat says she knows. Okay, so we have here the connection established. So if we don't have further questions here, we have one question by Monica Silva. We have a question from her saying, do you ever find MATLAB useful in your image analysis?
MATLAB is an extraordinary powerful tool for image analysis, and most image analysts that come from bioinformatics background use MATLAB. So since I come from a biology background, I'm one of those lazy image analysts that just uses point and click tools. So I showed you some of the analysis that I did with, for example, for Fiji and for Elastic. I am not an expert in both of those. I am what you could call an advanced beginner. I know a few things, but there is a lot more to learn. Since I come, this is a mea culpa. Since I come from a biology background, I tend to favor tools whose language I can understand better. So I'm taking my first steps in programming right now. And so I come from a different approach, and I am not using MATLAB or R packages or something like that yet, but it is definitely a very powerful tool. I'm going through side roads right now.
Thank you, Luís. And the keyword is really yet. So in the future, you will also use these and other tools. So thank you, Luís. Thank you. I think we are approaching the end of this sessions, the time for the end of this session. And if you have further questions in the future, you can contact Luís. And I would like to start by thanking Luís again for accepting our invitation to give this talk. Thanking Professor Solveig for also being here with us and sharing her thoughts and presenting Luís. And thanking Mariana Marques, who is here on the backstage and ensuring that everything goes well. And thanking everybody that attended this session. Thank you for your presence, and stay tuned for the next editions of the CNC meetings and other events from